Greetings. So primary literature always follows the same kind of format and it has the same elements. So any paper that you read that's primary literature, reporting on how a study was conducted and its findings, it's going to follow this template. First, you're going to see the abstract, and that's just like a one-paragraph report or summary of the literature. Then you're going to have a section that addresses an introduction and background. That section is just going to kind of tell you where the field is at a whole, what topic is being explored in this, in this primary research study, where the gap in that field is, and then how this study contributes to that gap or helps kind of advance the field. So it's basically just going to give you a brief orientation, the information that you need to know to be able to understand why the study was being done and why the findings are important. Then you're going to get to a methods section. The methods section is going to be pretty technical, and a lot of times, for undergraduate students, you maybe don't need to spend so much time reading through the methods section. In psychedelic research, sometimes you do. There are some elements that are pretty important there, but it's just kind of important to know that's what's going to be in that section. They're really reporting on the protocol that was used for the study, the methods that were used for the study, how that study was conducted. On the research side, when we're writing a methods section, we want to kind of think you want to give enough information so if someone else in another lab wanted to replicate this study, they could. We also think you want to give enough information in the methods section so that someone who's reading your study can understand what you did and they can use that to make their own interpretation of your findings. So that's the methods section. Then we're going to see the results section. Now, in the results section of a paper, we're just reporting what we found, the results, the raw data. We're not making any interpretations on those results. We're not saying what we think they mean. We're just saying what we found. So anyone can read that, and knowing the methods that we use, they can make their own interpretations of what those results mean. Then in the discussion section is where we discuss what we make of those results. Now, for us in research, the discussion section is really kind of the area where we can express our own independent thinking skills, our own creativity, things like this. And oftentimes, discussion sections also follow a bit of a template or a format. First, you'll have a summary of the results and kind of the study as a whole. Then you'll usually have a section that addresses novelty and innovation. So what is new about the findings that were observed in this study, and how is the study or the methods or the findings innovative? What about this work is innovative? What about this work helps advance the field? Because in research, everything that we do in research relies on funding. Usually it's federal funding, which means that we're trying not to repeat anything that has already been done, because that's not going to be a good use of federal funding, right? So we're really trying to, and this is what's addressed in that intro background section, we're really trying to have a good understanding of the field that we work in and identify where the needs are, where the gaps are in that field. And then we kind of come in and we try to help contribute to those gaps and advance the field or innovate the field. So that this section in the discussion section is really going to address that. Then we move on and we talk about how the findings that we observed relate to existing literature. So we might make an observation in our results, and this is where we might say, this other lab has also observed something similar to this, or this other lab observed results that are completely opposite of what we observed, or no one has seen anything like this, or something like this, so we just kind of place our findings in the broader scope of the literature. Then we're going to usually have a section on clinical implications. So how do our findings relate to clinical care? How can clinicians, if they read this, take our findings and apply them? What does that mean for clinical care? Things like this. And then at the end, we'll have always a section that addresses limitations and strengths. So in research, we actually are very humble and a very pragmatic field. It all depends on funding, and funding is often very limited. So usually we kind of think, in an ideal world, I would have buku bucks to fund this fully powered randomized controlled trial. I would have 4 different arms. I'd control for all the variables. And then in reality, I don't have unlimited funding. In reality, maybe I have a small amount of funding. In reality, maybe it's really difficult to recruit participants for my study. So rather than having 4 arms, we're just going to have 1 arm, and we're going to look at participant scores before they enroll in the study, and that's going to be the control, right? So we make these modifications. So this part of the discussion section is where we can really address what those limitations are. It's also where we can really set the scope of our work. So we can kind of discuss, let's say this study was conducted in individuals who have, very severe depression and suicidality. And in this population, here's what we found. But we excluded anyone with a comorbidity. So anyone who had very severe depression and anxiety, we excluded them. If they were already taking medications, we excluded them. So we kind of go through that. And based on that, we might say, these findings so far, really, you can only apply them to individuals who have very severe depression with suicidality without any other coexisting diagnoses or mental health issues, right? We haven't tested that in these people, so we don't know how our findings would generalize. And then that kind of leads into the future research section, which I don't have bulleted here, but that is usually another section. That's where we would say the next step would be to test this in a broader population. In research, we just accept there's always going to be limitations in our studies. That's life, that's pragmatic. And we actually tend to look at that as a good thing because that kind of shows us where our next step needs to be. And then, of course, also we address the strengths of our study. Here's where our study was unique. Here's what's strong about our study, things like this. And then again, that section of next future implications. So if we skip back over, that was the discussion section. Then we're going to see a conclusion section, which is usually just kind of an overall 1-paragraph summary of the study and what the main takeaways are. And then we're going to see this little section that usually has formality statements. There might be appendices in here. You're going to see a paragraph on author contributions. So who did what, who contributed what and how to the study. There'll be a section on financial disclosures. So is anyone receiving funding from another company, for example? If you're looking at studies that are conducted in animals, there'll usually be a paragraph on how animal rights were protected. If we're looking at genetic studies, there will be a paragraph on genetic information and how that was protected, cell culture, same thing, etc. So this kind of ancillary, again, formality statements. That's going to help you remember and really lock in that information. Take shorthand notes, abbreviate, right? Definitely abbreviate. And those abbreviations are actually going to help you learn a little bit better as well.